So I think as you guys have already heard, we're um, very interested in what Timothy's work in Kelly Fraser's lab, who's also on the call today, has to share with us today. We've already been talking about different groups growing many different lines in different contexts. As I mentioned earlier, when there were just a few of us on the call, two different talks, at least in this series, one from Nicolo Caporale in Giuseppe Testa's group, and one from Anton Balanos in Paula Arlota's lab. Also, Faravelli is the first author on that one, growing many organoids together. But the way that Timothy's growing cells is very different. Many more lines, and also with a more sort of general genetic interest than we've been concentrating quite a lot on neural lineage in this series. So we're really happy to have you on. Thanks for coming. And can you steal the screen for me, or do I need to stop sharing first?
I think you may need to stop sharing.
Okay, take it away. Is that working?
All right, you guys can see my screen now, right? My presentation?
Yes.
Okay, excellent.
Great.
Well, thank you for the invitation to give this talk, Carlo. My name's Tim Arthur. I'm a PhD student, a late stage PhD student. My defense is scheduled in just 2 months. So,
oh, nice.
I'm in Kelly Fraser's lab, who is also on this seminar call. And today I'll be talking about a couple of projects that we've been doing with the iPSCORE collection. And at the end of the talk, I'll discuss how to access the data and where it's deposited and all that kind of stuff. So the iPSC Omics REsource, or iPSCORE, is a large biorepository of iPSCs with multi-omic characterization. It consists of 3 tissues: the iPSCs themselves, and derived cardiovascular progenitor cells and pancreatic progenitor cells, which I'll refer to as CBPCs and PPCs throughout this talk. Over 1,000 molecular samples have been generated on these cells, and there are over 221 iPSCORE donors. Of the molecular data types we have whole genome sequencing, RNA-seq, and ATAC-seq, and H3K27 acetylation ChIP-seq, as well as others that I won't really talk about today. iPSCORE has been very fruitful for the Fraser Lab. There have been over 20 publications over the past decade that have risen from data generated from iPSCORE, and today I'll touch on 2 that I am the first author on. So first, I'll be talking about mapping multi-omic QTLs in stem cells and derived tissues. Next, I'll be discussing a paper that's published in Nature Communications that kind of investigates variability across iPSC cell lines based on pluripotent cell state regulatory networks. And finally, I'll discuss data access and availability. So this first section was co-led by myself and Dr. Jennifer Wen, who recently graduated from the lab and is now a project scientist at UC Irvine. And our motivation for doing this project is that molecular mechanisms underlying complex diseases are poorly understood. This is because complex diseases are driven by have a lot of polygenicity, which implicates many genes, and their associations are also often difficult to disentangle. And then there's this level of incomplete penetrance, which also blurs the role of genetics in the pathology of these pathology and heritability of these diseases. So the primary way that genetics are investigated for complex diseases is using genome-wide association studies. Essentially, a linear model is used to calculate the association between all genetic variants across the genome and a disease or a trait phenotype. In this example, there are 3 loci, this toy example, there are 3 loci one on chromosome 6, one on 8, and one on 13 that are above genome-wide significance that are associated with heart failure, let's say. These results have been historically difficult to interpret because over 90% of the genetic variants associated with these diseases are in non-coding and intergenic regions of the genome, which makes functional annotation of their role difficult because there's no context of which gene is affected. Quantitative trait loci, or QTLs, enable the functional characterization of GWAS, and the QTLs are essentially genetic variants that are associated with a molecular phenotype, such as gene expression. In this toy example, we have variant A that is associated with lower expression of this gene, and variant G, which is associated with a higher expression. You can overlap these eQTLs with GWAS loci and determine whether or not this gene is underlying a GWAS signal. This approach has been very helpful, but it has only explained 43% of the GWAS loci across traits. So additional information is needed to kind of characterize the majority of GWAS loci that remain annotated by eQTLs. One approach is to integrate multi-omic QTLs, which affect different molecular phenotypes. And this can identify GWAS loci that aren't associated with an eQTL. And specifically, we can look at chromatin QTLs like CA QTLs, which affect the chromatin accessibility, which is assayed by a TAC-seq, or HA QTLs, which affect histone acetylation, which is assayed by H3K27 acetylation. And here we have the same Manhattan plot in the toy example, and you can see that this loci is theoretically associated with a eQTL, and this loci is associated with a CA QTL. So layering on this additional information and integrating epigenomic phenotypes can help characterize these these loci without any associations with gene expression. So to do this, we analyzed 1,261 molecular samples from the iPSCORE collection, primarily focusing on epigenomic samples that haven't been released in previous studies. There were 393 that are indicated by the asterisks in this table that are newly released from this study. We characterized to assess the quality of our TAC-seq and epigenomic data across these iPSCORE tissues. We performed a UMAP analysis based on chromatin accessibility and saw tissue-specific clustering, indicating that these samples are distinct from one another. And we next wanted to see whether or not their epigenomic properties are kind of aligned with their mature tissue counterparts. It's well known that iPSC-derived tissues are immature relative to their adult counterparts. So it's always important to ensure that the epigenomic properties are comparable between their counterparts. And we see that iPSC-specific TAC-seq peaks are enriched with binding sites for transcription factors that are involved in self-renewal and pluripotency, like NANOG and OCT4 and SOX2. CVPC-specific TAC-seq peaks are enriched with binding sites for NKX2-5 and MF2C, which are known cardiac markers. And PPCs are enriched for pancreatic markers like NKX 6-1 and PDX1. So taken together, this shows that not only that our TAC-seq data is good, but they are representative of their adult counterparts. So we sought to characterize map the QTLs across these 8 multi-omic data sets. We applied a linear mixed model, which accounts for relatedness and kinship of individuals within the iPSCORE consortium. And we calculated up to 3 conditional QTLs to account for multiple loci associated with a given gene or a taxi peak or chip peak. And this approach identified 70,218 QTLs across the data sets, 60,000 of which were primary, and 10,000 of which were conditional. So they were these were for elements that had multiple QTLs. Interestingly, we saw that about 50% of the expressed genes in iPSCs had an eQTL. This could be related to the sample size being the largest across the 8 data sets, and the fact that the cell type is more homogeneous than the CVPCs and PPCs. It also is a remarkable finding because it shows that there is a lot of regulatory variation that affects gene expression in stem cells. We also found that there are threefold higher HA-QTLs than CA-QTLs in CVPCs. You can see that there are only 5% of ataxia peaks in the cardiac cells have a CA-QTL, and 15% of the ChIP-seq peaks in the cardiac cells have an HA-QTL. So this is a good indication that maybe ChIP-seq is a good molecular phenotype to investigate for trying to identify regulatory variation. So after identifying these QTLs, we sought to characterize their properties, and we looked at three annotations to characterize them further. The first is chromatin state. Are these QTLs enriched in cis-regulatory elements like active promoters and enhancers? Next, we characterize their complexity. Do QTLs affect multiple molecular phenotypes? For example, does a single QTL affect the expression of a gene and the accessibility of an ataxia peak? And then finally, we look at stage specificity. Are the QTLs active at different stages of life, or are they shared across multiple stages? So chromatin state, we annotated the QTLs using chromatin states determined by the chromHMM in the EpiMap repository. And we saw, consistent with previous findings, we saw that eQTLs were primarily enriched in promoters and had lower enrichments in enhancers and transcribed regions, where the chromatin QTLs, CA-QTLs, and HA-QTLs were primarily enriched in promoters and enhancers. You could see that there's a slight increased enrichment of HA-QTLs and enhancers, and this is likely because H3K27 acetylation is primarily a marker of active enhancers over promoters. To characterize the QTL complexity, we asked whether or not QTLs can affect multiple molecular phenotypes. As I mentioned earlier, a single QTL can affect the expression of a gene and the accessibility of a peak, or the accessibility of an ataxia peak and a ChIP-seq peak. So to do this, for each tissue, we measured the LD between lead variants from the QTLs of adjacent QTLs. And we used a network-based approach to identify roughly 5,600 modules of QTLs affecting multiple elements. And these modules were composed of QTLs that affected between 2 and 15 different elements. And you could see that the majority of modules were composed of QTLs that only affected 2. So focusing just on the cardiac cells, since it's the most complete data set that we're working with, we see that 51% of the QTL modules are composed of at least one CA-QTL and one HA-QTL. And this is probably supporting the concordance of chip peaks and ataxic peaks, both capturing enhancers and promoters. In total, of all of the QTLs, we saw that 22% were within a module, and about 80% were singletons and only affected one molecular element. So let's see. Next, we wanted to characterize the stage specificity of our QTLs, and it's well known that iPSC-derived tissues are immature and resemble fetal tissues, and they can provide an important insight into this underexplored developmental interval. And the underlying biological principle governing temporal regulatory variation is that biological processes or regulatory networks underlying biological processes in mature and developing tissues are different and can be mediated by different transcription factors. So in the adult tissue, we see this transcription factor is regulating the expression of this gene, and this variant can affect the binding of this peak, of this transcription factor, and the variant affecting the binding of this transcription factor that's active during fetal development can be distinct from the mature or the adult-specific variant. So to calculate stage specificity of our eQTLs, we define two developmental stages using adult eQTLs from 47 GTEx tissues. I know this may not be this crowd might not be familiar with GTEx, but it's this large collection of adult tissues obtained from 44 different organs from hundreds of people. And it's one of the largest eQTL databases available. So we used eQTLs from GTEx to define the adult stage, and the early developmental stage was defined by eQTLs discovered in this study from the 3 iPSCORE tissues. We applied MASH-R, which is a tool that is typically used for identifying cell type specificity of regulatory variation, to about 100,000 eQTLs from these two databases, and identified 1,237 EDEV-specific eQTLs, which is just a small fraction of all of the eQTLs that we've discovered in our study. The vast majority of the eQTLs that we found were shared between the adult stage and the early developmental stage. We calculated the correlation to validate that these are truly temporal. We looked at their activity in both iPSCORE tissues and GTEx tissues, and we saw that the temporal regulation, including the EDEV-specific and adult-specific, were not correlated between the two data sets, where the shared eQTLs were correlated between iPSCORE tissues and adult tissues, indicating that they're active at both the early developmental stage and the adult stage. We further characterize these EDEV-specific eQTLs by looking at the distance of the lead variant relative to the gene that it affects, and found that EDEV-specific eQTLs are more distal from their associated E gene. And we also looked at whether or not their effects were smaller than shared eQTLs. Or we compared the effect sizes between EDEV-specific and shared eQTLs and found that EDEV specific had smaller effects. So we wanted to integrate these QTL characterizations into understanding disease-associated loci. So we co-localized our iPSCORE QTLs with 16 GWAS for tissue-relevant traits and disease. Some of the cardiac traits that we looked at were myocardial infarction and QR restoration, pancreatic and metabolic traits like type 2 diabetes, and fasting glucose, and some developmental traits like birth weight and childhood obesity. And then we annotated the co-localized GWAS with QTL annotations that we defined above, previously, like molecular phenotype complexity and stage specificity. So here we show the number of GWAS loci that are co-localized with these different QTLs. We first calculated the independence of GWAS loci within a given trait and identified 5,200 independent loci across all 16 studies. And we determined whether or not these loci were co-localized with a QTL from our study, and identified about 10% of them were associated with an iPSCORE QTL. You'll notice that by the large proportion of this graph that's taken up by red and light purple, which are representative of CA QTLs and HA QTLs that co-localize with the GWAS loci. Integrating chromatin QTLs can explain a lot more GWAS loci than compared to just eQTLs alone. So layering in multi-omic QTLs would explain some of those unannotated some of those GWAS loci that are not annotated by affecting gene expression. And we wanted to see whether or not complex QTLs were more likely to co-localize or explain a GWAS loci. So we calculated the relative enrichment of GWAS co-localization based on the composition of modules. And we found that modules consisting of CA QTLs and HA QTLs, or chromatin QTL associated modules, were the most enriched for co-localizing with GWAS. And in general, modules were more likely to co-localize with GWAS compared to singleton QTLs. So adding on this layer of information about chromatin accessibility and epigenomic properties can provide more functional information on a GWAS loci, and it can also identify more GWAS, as I mentioned earlier. Surprisingly, we only found 33 EDEV-specific eQTLs, which I described earlier, that co-localized with a GWAS locus. And this represents a strong depletion relative to shared eQTLs. And it suggests that regulatory variation that's active at multiple stages of life or throughout life is more responsible for explaining disease heritability for complex traits. We found an interesting complex QTL module that is associated with a QRS duration locus. QRS is a part of an EKG, and it describes the electrical impulse of the heart. And this CVPC QTL module, which is composed of 3 ChIP-seq peaks and an ATAC-seq peak, co-localizes with a Q restoration loci that's located within the intron of KLF12, which encodes a transcription factor. KLF12 has previously been associated with Q restoration, but has not been associated with the expression of the gene itself. We sought to provide more functional information on how this loci might be working. We sought to further characterize the molecular mechanism for this locus. So we intersected the significant variance from the GWAS and overlapped it with the transcription factor binding sites in the ATAC-seq peak. And we found that one of the variants for significant variance for Q restoration overlapped a binding site for cardiac specific transcription factors MEF2A and MEF2C. I've highlighted where the variant falls within the binding site. So this can provide information and the framework to do functional validation to help explain why this loci is associated with Q restoration. So to conclude the first section, this is one of the largest multi-omic QTL studies to date with paired molecular samples. We used over 1,000 samples from iPSCORE to do this analysis. And we showed that a large fraction of QTLs affect multiple elements. And we also demonstrate that integrating multi-omic QTLs can help explain more GWAS loci. We also found a modest number of GWAS loci that co-localize with EDEB-specific eQTLs, indicating that adult-specific or shared eQTLs are more responsible for disease heritability. And this paper is currently in revision at Cell Genomics and available on bioRxiv. If you're interested, here's a QR code if you want to take a look at the preprint. So with that, I think maybe I can stop and take questions on the first section, or I can continue. What do you think, Carlo?
Yeah, I think that's a great idea. Does anybody have any questions? I know I do. I'll wait for a second to see if people want to jump in.
I have a question about the GTEx data that you showed.
Sure.
So you know, we looked at GTEx data quite extensively, and one of the things that we are interested in is looking at the multi-tissue QTLs and single tissue QTLs that they talk about.
Mm-hmm.
And I want to know if you have taken a look at them separately in your data to see if they show different patterns.
So we don't consider, we just aggregate all of the lead variants from GTEx and consider them adult specific. We don't really look at tissue sharing across GTEx for determining stage specificity.
Okay.
Yeah.
I thought it would be interesting since you have like the cardiac cells and, you know, two different differentiated cells. It could be interesting if you have that tissue specific eQTLs and see if, you know, if you see a specific pattern specific to the tissue eQTLs. But
Yeah, that's a good point. So kind of pair the CBPCs with the adult tissue counterparts and characterize them in that way is what you're suggesting?
yeah, exactly.
Yeah, that would be interesting.
And I have a second question. It's short. What's your plan for expanding the HA QTLs? Because, you know, you're looking at a single histone acetylation mark, I'm understanding, right? So do you have plan to expand it to like K4 trimethylation, 27 trimethylation, or any other markers? Yeah,
this is actually something that Kelly was briefly considering, but we're collaborating with Graham Nick Vicker, who has extensive experience in both genetics and looking at these epigenomic marks. And I believe he was part of a team that tried to do QTLs on some of these other histone markers, and he said by far H3K27 acetylation was the least noisy, I guess, and yielded the best results for identifying these regulatory variants. I think the signals for H3K27 acetylation tend to be a little bit stronger than other histone marks. I could be wrong, and that really affects the power to detect these QTLs. So we don't have any plans for extending this into different chromatin marks, but it would be incredibly interesting.
Yeah, that's interesting. Thank you.
It looks like Aditi Sundaresh from Helena Kilpinnen's lab also has a question. Jump on in, Aditi.
Hey, and hey, Tim, thanks for the talk so far. I'm a bit out of my depth here, so I'm sorry if this is a bit of a naive question, but I was just thinking kind of one of the last statements you had was about the EDEV specific EQTLs not really accounting for as much of the GWAS hits. But I'm just wondering, the GWAS data that you have, do you know what the age distribution of those samples is like? You know, is it possible that they're really coming mostly from adult populations? And so you don't have the data that captures what would be affecting a younger population dynamic, and that's why you're not accounting for them with the EDEV EQTLs.
Yeah, so I think this the genotype of the stem cells that we have are influenced by reprogramming, but they're essentially reflective of the genotypes of the people that they're derived from. So the genetics that shouldn't change much other than somatic mutations that are that arise from the reprogramming process. So
can I jump in, Tim, a little bit too?
Go ahead. Yep.
So I think your question is actually spot on. And what we should be saying is that these are GWAS of adult traits. All right. So regulatory variants that are active. So one of the big questions in the field of, you know, what's missing variation in GWAS is it active in an earlier developmental state? Right. So people throw out, okay, you know, you can imagine if you made more beta cells in development, that as an adult, you'd be less likely to be susceptible to type 2 diabetes. Just a trivial, like a straightforward example. But what we're finding is that regulatory variants that are active in only the early developmental stage, well, they can contribute to adult phenotypes. That's not where they're depleted for doing so. And that's probably not where all this missing variation is. That's not to say that regulatory variants that are active in the fetus are not contributing to phenotypic differences that are occurring during development and at birth.
Yeah, well said, Kelly. Thank you.
Thanks.
So I have a question that kind of intertwines with that one. Could you rewind to your slide 19? I thought it was super interesting. So here you show that the early specific QTLs are of smaller size.
Mm-hmm.
But at the same time, and I think maybe the slide before this, you show that the vast majority of signals are actually both, they're shared right there in the, so the slice you're looking at in early specific is kind of a very rarefied piece of this.
Mm-hmm.
I was wondering if you slice the data by looking at the shared QTLs, but looking at the effect sizes separately in the early tissues and the iPS-derived tissues versus the adult tissues. And the reason I asked the question, I don't know if you guys have seen this. I think it's still in bioRxiv. I don't think it's published yet. But Mike Gandel and Luis Torre Ubieta did a brain-specific EQTL study. I think they had somewhere in the range of like 600 fetal brains. They did EQTL analysis, and of the EQTLs they found, the effect sizes are actually bigger in fetal life than they are in adult life. But those are, of course, of these, to even make that comparison, it has to be a shared QTL. So I'm wondering if the small effect size you're seeing in the early tissues is reversed if you look at the shared QTLs. Because
Yeah.
their result would actually suggest that the effect size might be bigger earlier.
Yeah, I think maybe Mike Gandel is reviewer number 4. Thanks for sharing that. Yeah, I'm not quite sure how to reconcile that. In the paper that's on bioRxiv, we tried to look at evolutionary constraint as defined by PLI scores based on which genes are affected by fetal EDEB specific and shared EQTLs. And we saw that EDEB specific E genes tended to have higher constraint, therefore might tolerate EDEBs might tolerate regulatory variation with smaller effects. But that was something that didn't fly with reviewer number 3 or 4 or whatever. So we've actually taken that out of the paper, and we're still trying to reconcile this with what was observed in that paper. I don't know, Kelly, do you have any insight?
No.
Yeah, could you, if you look at the 19 again, could you just split just look at all the shared EQTLs, but then just look at the effect in your early tissues versus the GTEx?
We could do that, but one thing that is challenging is the difference in sample sizes, which has a direct effect on the effect sizes, comparing effect sizes across tissues.
Well, it should affect the significance. It shouldn't really impact it might impact the stability of them, but it shouldn't systematically bias the effect size, should it? Or is that a miss?
I think it would have an effect on the effect size. And I guess, well, it would have an effect on the, I guess, error
Right. So don't look at the effect size, not the
Right.
effect size divided by variance.
Yeah. Okay. Yeah, that's a good idea. I can look into that.
Because it could reverse the whole thing, right? I don't know, because I guess I'll stop after this. But the fact that there's so few early specific QTLs makes me think they're of some different class that's not involved in the Gandel observation. Because Gandel only observes things that are in both the fetal and the adult, because that's how he says they have a larger effect size in the fetus. So that would be the slice of your big shared bunch. And the small effect size you see in the early specific could just be like a different flavor. But anyway,
Yeah.
I'll let us proceed to any more questions, or if there are no more questions, to your second topic.
Yeah.
Does anybody want to jump in?
I mean, I will follow up for a minute on those comments, and that is that just for the edification of the group, that it's complicated because a gene has multiple regulatory elements. I mean, this is the classic regulation of hemoglobin, right? So some elements are shared between the different stages. Some elements are only active in one or the other developmental stage. And so if just because it's unclear what it means that the developmental specific EQTLs have lower effect sizes, because there could be shared that overpower them, but they could be just tweaking it a little bit. It's not clear what it means in development. I would say it's more of an observation than concrete understanding.
Gotcha.
And all of these studies, there's power issues, right? So if you look at the data one way, you can see something. If you look at it another way, you can see something. So I understand what the concern of reviewer number 4 was. And I think that we have addressed it. And although it dampened down the message of the paper, perhaps it's more accurate.
Okay, we're running out of time. So I
But
will.
this group may be really interested in this, so try to go fast because you know this really well. So go fast.
Yeah, and this section is shorter, so it should be fairly
Great.
quick. I think I can do it in 10 minutes. So this paper is published in Nature Communications if you're interested and want to follow up. And it's essentially looking at the variability of pluripotent cell states across many lines. As I highlighted earlier, we have over 200 lines of iPSCs. So this enabled us to look at things like the primed state, the proportion of the prime state and formative state across all of our lines. So we wanted to generate gene networks for across using these lines. So we calculated the global gene co-expression using 221 iPSC RNA-seq samples and used an unsupervised network approach to identify these modules of genes that are associated with each other. We used external data sets, external gene sets to perform enrichment of genes within these gene network modules or GNMs. And we found that GNM5, where is it? GNM5 is enriched with epiblast and naive gene sets, while GNM10 is primarily enriched in the primed gene set. We generated paired attack-seq for these samples for 142 of these lines, and regulatory networks should underlie the association between genes across the genome and should be bound by the same transcription factors. So we performed the same unsupervised network approach to identify modules of associated attack-seq peaks and found 13 of these modules. And we did something called cellular deconvolution using an external data set of differentially accessible attack-seq peaks in formative versus primed cells, and used this to estimate the relative proportion of the formative state across our 142 iPSC lines. And we see that the majority of the cell lines are mostly in the prime state, which is consistent with previous observations. And there was a range of 0 to 32% of cells in the formative state across these 142 lines. So each bar, this is a series of 142 bars, and each bar represents a single line, and the y-axis is the estimated cell state proportion. We calculated a score for each of the regulatory network modules and ran a linear model to determine whether or not the estimated formative proportion as calculated in the previous slide is associated with the accessibility of these regulatory network modules, and found 3, RNM2, 3, and 8 associated with the formative cell state proportion. This is a very complex figure, but I'm going to focus on the 3 RNMs here and here. We characterized the epigenomic profiles of the ataxic peaks in these RNMs using external cell state annotations, chromatin states, and transcription factor binding sites. And the 3 RNMs associated with the formative state were tended to be enriched in promoters. 2 of them were enriched in enhancers, and 1 of them was enriched in bivalent chromatin and active promoters. RNM3 is interesting because it's exclusively enriched with binding sites of the TEAD family of transcription factors, and RNM2 is enriched in the kind of canonical pluripotency transcription factors like NANOG and OCT4 and OCT2. And RNM8, which is associated with bivalent chromatin, is associated with a lot of transcription factors associated with cell cycle regulation. I wanted to focus on RNM3 because it's kind of a distinct regulatory network within the naive state. And previous groups have shown that if an iPSC is in a naive-like state, you can differentiate it into extra embryonic lineages. And so I intersected our ataxic peaks with cell type specific ataxic peaks from the Descartes data set from fetal tissue, and found that ataxic peaks associated with the extra embryonic lineages were enriched for overlapping with RNM3. So we felt that this can kind of improve the differentiation protocols for placental and neuronal lineages. And it's consistent with TEAD4 kind of specifying the lineage commitment to the trophectoderm. Again, this is a published paper. It's on Nature Communications, and the conclusions are that pluripotent cell state proportions vary across iPSC lines. There are distinct regulatory networks mediated by TEAD and NANOG OCT4 that are active during formative pluripotency. And TEAD RNM3, it should be, is enriched in ataxic peaks from extra embryonic lineages and placental precursor lineages. So I'll spend 2 minutes just explaining where this data is and how to obtain it. There has been a monumental effort from Jennifer, who I mentioned was a co-author on the first project, and a research associate in the lab, Ben, to make this data publicly available. Data from previous studies are available on dbGaP as version 4, and we're currently uploading approximately 1,500 additional samples, which includes data from every single study that has come out of iPSCORE, and it will have single nuclei ataxic peak, bulk RNA, single cell RNA, seq, Hi-C, pretty much anything, any molecular data type that exists is going to be deposited in version 5. So
I would also like to mention that the iPSC lines, you can order them, like if you only want 2 or 3, you can get them from YCell. But if you want the entire collection of the iPSC lines, we have them organized in a manner that they're easy to ship. And so we've distributed them to 3 or 4 different academic groups so far, and we're happy to distribute them to 3 or 4 more.
Great.
Yeah, and the last is just acknowledgements. So want to thank Kelly and Jennifer and Matteo, who was a former postdoc, now a PI at UCSD, and Martin Pera, who was really instrumental in that second project that I rushed through. But with that, I'll leave it open for questions or discussion.
Great. Questions? Thanks so much. I had a quick one at the end. You list dbGaP where everything is. Do you guys have a place where you put sort of process tables under non-controlled access, or is it all there in dbGaP?
So there are individual fig shares that are associated with the different publications. But
we also have so the dbGaP is like 99.9% done, but you know, it's like the last 0.1% that's always a pain in the butt when you're dealing with dbGaP. So we have one more summary statistics that we have to send them, and then they should be able to release all the data. But on my website, on the Fraser Lab website, there is pages, and there's one more page that's going up, which is going to describe the data availability in absolute detail. And I'm hoping that the dbGaP is finalized in a week or two, maybe 2 or 3, to be honest. And then that last page will go up and it will be very clear. But there's on Figshare, there will be a single, there's different studies for Figshare, but there's also going to be a single Figshare set where you can get all the summary statistics.
Great. Great.
But it should be easy. It should be easy to download the data from dbGaP. I mean, dealing with dbGaP is more of a pain on uploading the data. Downloading the data should be easy to get.
Okay.
And if you are looking for something specific, you can reach out to us and we can provide it too.
Thanks. Any questions about the second half, guys?
I have a question. It's not so much a question as a sort of comment and leads into a question. And thanks so much for presenting all of this. It's really great to talk to people who are interested in this problem. And it seems this is a sort of way of saying, I mean, I have so many issues that I want to discuss with you that I can't possibly raise them all now. So thanks so much for organizing this kind of session. And maybe we should look at ways of talking to each other as a group more in the future or as individuals. But at any rate, maybe I could be more specific and ask you about the last image you showed from the section 2 of your talk. That one, yeah. Where you talked about neurons. Is it the next slide? Or this one? Yeah, what were you saying here? I just want to be clear what you're saying. RNM3 is associated with a kind of a trophoblast type of signal, right? Is that correct? That's what I understand. Yagi et al. Is a Teed paper, if I remember. Right. So then go for it. Just fill me in. What's going on here?
Yeah.
The neurons are RNM2 and RNM8 is a retina.
So
is that ganglion and photoreceptor?
Yeah. So I rushed through this, but essentially I annotated our iPSC peaks. So these are ataxic peaks that are present in the stem cell with cell type specific peaks from the Descartes data set, which has, I believe it's like 30 or 40 different fetal cell types from fetal tissue. So these are iPSC peaks annotated by fetal specific cell type peaks. And then I calculated the relative enrichment across the RNMs. So what I'm highlighting here is that there are ataxic peaks that become active in trophoblast differentiation that are in RNM3, which is primarily enriched with TEAD4 transcription factor binding sites. And then RNM2 is enriched for ataxic peaks that become active during the development of these neural lineages. And RNM2 is primarily enriched with nanog and OCT4. So there might be these two independent regulatory networks that are active during naive pluripotency that can give rise to different lineages at different capacities. Or yeah, give rise.
That's great. Thank you.
One of the overarching things is that when you think of, we have 220 or so or plus different iPSC lines. And it's been known for a long time that an iPSC line has different types and different proportions of cells in different pluripotency states. So some will be more prime, some will be in the more formative, some will be more in the naive state. And so, I mean, people who differentiate iPSCs know that some lines are really good at becoming neurons and other lines are really good at becoming something else, right? And what Tim has shown is that each line, and it seems to be stable, each line has different proportions of different cells in these pluripotency states. And the different cell states have different active networks. And so the networks that are active in the naive state are the ones that are more likely to give rise or overlap what's present in neurons. And so those iPSCs that have a larger fraction of cells in that particular cell state are probably more likely just to differentiate more readily into neurons. So somebody's asking about.
Do you know that actually? Sorry to interrupt, but for example, could the difference between 3 and 2 be explained by SOX2 so that in 3, SOX2 is active with T, but in 2, SOX2 is active with nanog and OCT4. And I'm just, there we go.
Yeah, so SOX2 has high sequence similarity to left1, so it was lumped into the same group. And it's enriched in RNM2.
They bind the same transcription factor site, so you can't distinguish them.
Yeah, the sequence between the binding sites are indistinguishable.
No, because this is very cool. You know, Magdalena Zernicka-Goetz showed in the 4-cell or the 2-cell stage mouse embryo some very interesting work about the regulation of SOX2 and OCT4 and nanog. And I'm just wondering whether that would help us understand that image that you just showed us. And that to come back to the specific point you were making, Kelly, I think what you're saying is very important if that's true. And it would be really nice to sort of think more about this. I hesitate to direct everybody's attention in this direction. But thank you, that was helpful.
Yeah, but do you know Martin Pera?
Yeah, sure.
Yeah. We worked with him closely on this, and he just loved this project. So this is actually kind of, the interesting thing about iPSCORE is it's taken me in all types of different directions because we generated this data and then we were like, okay, how can we analyze it, right? Because it was time consuming and costly to generate it. And so we've done all types of things, and many things that we've done, I do genetics, and the whole goal was to generate all this data to look at genetics. And the first talk that Tim gave you was all on looking at the genetics. But although there was some genetics in this second presentation he gave, the genetics is subtle, and it's really would you say that's true, Tim, that the genetics may be playing some role in helping define the different proportions of cell states, but pluripotency cell states, but overwhelmingly, it's probably more a byproduct of the reprogramming.
Yeah, I would.
Tim, I'm gonna let you jump in.
Yeah, I would say that in general, environmental stimuli have more of a substantial effect on phenotypes than genetics. you know, in complex traits at least, such as cell states in this case. So I think that culture conditions, specifically in this case, culture conditions is the most deterministic factor of how many.
Yeah, but the beauty of the way we generated the data is that the lines were all subjected to the same culture conditions. So culture conditions are known to change cell states, but here what we did is we held the culture conditions the same. So we were able to look at the different lines.
Right.
But still, it's probably more of a factor of the reprogramming, what happens to the epigenome during the reprogramming than it is genetics, why the different lines have different cell states. Although Tim did find some evidence of the genetics playing a role.
Yeah, I see that there's a question from Alexander.
Yeah, hi. Thank you very much. It's a really.
I have to run. I'm sorry. All right. Thank you.
Thanks for joining, Kelly.
Thank you. No, I'm just curious, like you say, exactly if you work in detail, like what kind of technical culture conditions have you found kind of could be associated with that? Have you tested like different culture, or have you done always the same homogeneous versions? Have you found any kind of technical, for example, to manipulate these states between cell line and or stabilities between passage and everything, like all the technicality of cell culture?
Yeah, that's a $1 million question that I think a lot of people are looking into.
Yeah, we're not developmental biologists. We're geneticists. So we held everything constant so that we could do genetics.
But you seem to think that genetics is still a minority of what you see. That's what I understood, right? It's not the larger effect.
I think culture can. These are all systematically cultured, but I do think minor variations in culture conditions can have a more dramatic effect than small effect genetic variants.
We have some work coming out in just a couple days that actually, speaking of Martin Pera, is being published on August 15th in his journal Stem Cell Reports, that actually almost looks at it from the opposite side of what stable expression traits in iPSC lines from, and multiple lines from the same individual can actually drive these stable biases. We weren't looking at single cell data, so we weren't looking at how many of different kinds of pluripotent cells there were, but we looked at biases in their transcriptome. So we can see actually transcriptome signatures that are specific to individual humans that bias their iPS cells differentiation propensities.
Yeah,
go for it. Go for it.
I think structural rearrangements and stuff that happens during reprogramming can probably have a large effect on an individual line's propensity for.
We've seen epigenetic effects of large size too. Yep.
Yeah.
So I have some papers I put on Slack, and I'll add yours. I had your first one, but not your second one. I'll add yours and ours on there, and maybe we can keep going here. There's one other question from Pao. If you want to unmute and jump in, Pao, go for it.
Yes, so I was wondering whether you have also tested whether the passage number of those lines alter the proportion of IPS states.
Yeah, so to calculate these RNMs, I ran a linear mixed model and included a bunch of covariates. Passage number was corrected for when these regulatory networks and gene modules were defined, basically. So that the effects of passage number should be controlled for in this, yeah.
But do you have like an idea of the extent of the variability that passage number can explain?
I did that. It didn't make it into the paper, and I didn't really see an association with passage number and the estimated formative cell state.
Very interesting. Thanks.
Just to go on that point for a minute, I mean, it's extremely interesting, this notion of passage number. what we found, I mean, there's something anecdotal about this, but some of the cells exhibit these different properties that Carla was mentioning in a remarkably stable way. Over a decade in different labs, analyzed with different tools. So while it may be influenced by some feature of the reprogramming, it's actually extremely interesting because it's so stable. So you can actually study it. And maybe as I said earlier, it's just fascinating. I don't really know where to
the
start.
existence of the H9 line was kind of a incidental benefit for us because this one ESL line that so many labs have used and so many times keeps doing the same thing no matter where it is or what it's doing. It's interesting.
Yeah, I think the genomics field is just starting to jump into iPSC biology. So it's an exciting time to be a computational biologist in this space because there's just so much to look at.
Thanks so much for joining us today. It was a really great discussion. And I'll post some more papers on the Slack channel and see if we can keep the discussion rolling. Really appreciate it, Tim. Good to meet you too.
Thank you, Carlo.
Likewise.
All right, bye.